Hello, मौसम. Hello, good morning, everybody. The topic for today's lecture is vertigo, the basic anatomy and physiology. I am Dr. Vishal Ansarajani, Associate Professor, ENT Department, Index Medical College and Research Center in Gaon. So, vertigo. Before uh, discussing vertigo, we need to discuss about the balance and posture. How do we maintain the balance and we are able to walk on two feet and also able to maintain the posture. So there are three components in our body that is required to maintain the balance and the posture. The first is sensing the environment. There are multiple sense organ by which we sense the environment that sends signals to the central nervous system, and the second part is the integration of these signals in central nervous system and creating an appropriate motor response for uh, correcting the balance and the posture. The third part is that performing of this motor activity and commands. Like we can maintain, if we are falling on one side, we sense that particular change in the posture by sense organs. The CNS integrates this all signals, creates a motor response, and the muscles of spine and lower limbs, shoulders, upper limbs, they perform this motor activity to correct the posture. Now, the first part is how do we sense the change in the posture for our body. The sense organ, there are three main sense organs that are continuously at work. The first is the vestibular system, which includes semicircular canals and otolith organs. The second one is visual system. They include the perceived movements, optokinetic movements, and saccadic movements. The third is somatosensory system, which involves or includes the pressure and a joint proprioception. So these three, vestibular, visual, and the somatosensory system, they send signals to the brain. And now we will discuss the vestibular system in details. The vestibular labyrinth is a part of the inner ear which is filled with fluid, and it has got two main functions. First is the hearing, which is judged by the movement of the fluid in the cochlea. And the other main function is the balance, which is by semicircular canals, utricle, and saccule. Now we'll discuss the main sensory cells of the inner ear. These are the hair, we call it as the hair cells. These hair cells, they detect the movement of the fluid filled in the membranous and the bony labyrinth. This fluid movement might be because of the different stimuli. The first stimuli will be your like sound wave pressure. If our ear is presented as sound wave, this will cause the movement of the inner ear fluids, and the it is judged by the hair cells present in the cochlea, and the further central nervous system judge that thing and respond accordingly. The another stimulation is the angular acceleration of the head, and that angular acceleration is judged by the semicircular canals and the linear acceleration. It can be a vertical or the horizontal acceleration, and it is judged by the utricle and saccule. This utricle and saccules, they are called otolith organs. Now we'll little bit discuss about the functions and how the hair cells function. Now we all know about the structure of the hair cell, like there is a cell body, and at one end there are some small cilia, we call it as a stereocilia, and at one end there is a large and a longer cilia which is called as the gynocilia. So this movement of the cilia is judged by the central nervous system by like an excitation or the inhibition. So depending on the direction of movement of this cilia decides whether this is an excitation or inhibition of. So these hair cells, they always have a resting neural firing rate. Like in this central picture, we can see this is a 90 spikes per second. It is a resting firing rate of, and this all the cilia and the are in the center. So whenever there is movement of stereocilia towards the kinocilium, towards the larger cilia, towards the kinocilium, there will be an increase in the firing rate of the hair cells. So this will be judged as an excitation, and whenever there is a movement of the stereocilia away from the kinocilium, this will be judged as the inhibition, and it will reduce the firing rate of the hair cell. Okay, so this is a basic fundamental. So stereocilia movement towards the kinocilium is excitation, away from the kinocilium is inhibition. So we'll discuss about the semicircular canal. So we all know that there are three fluid-filled canals, the superior one and the posterior one and the horizontal. These three fluid-filled canals, they are approximately 90 degrees to each other, and they represent the three dimensions, and actually they judge the angular movement of the head. There are three semicircular canals on each side, on the right as well as on the left side. So actually we have got six canals, three on the right and three on the left side, and they are in pairs in for the direction of the movements, in respect to the direction of the movement. Like lateral canal of the right side is coplanar in the same planar with the lateral canal of the left side. So lateral canals are coplanar with each other, and this lateral canal is 25 degrees backward, head 25 degrees backward tilts to the horizontal. The posterior canal of the right side is coplanar with the anterior canal of the left side and vice versa. So right and left posterior anterior canals are coplanars. Now we'll discuss about the sense organ. In the ampulla of the semicircular canal. Each semicircular canal has got one dilated end, and the dilated end is called the ampulla. Okay. So then each ampulla, this picture shows the structure of the ampulla. This is all ampulla, and at the center, this ampulla has got a mount of hair cells, which is known as the crista ampullaris, and from this crista ampullaris to the opposite wall, there will be a fan-like divider. This divider is called as the cupula, and this hair cell, the cilia of these hair cells are freely lying into the cupula. So whenever there is a movement of the endolymph, there will be a movement of the cupula, and the cilia of the hair cells in the same direction. Now, the function of the semicircular canal. So, semicircular canals, they judge the angular acceleration of head. And how does it happen? Like here in this picture, we can see this is the lateral semicircular canal of the right side, and this is lateral semicircular canal of the left side. And on the anterior aspect, there is a dilated and we call it of the lateral semicircular canal. It is the ampulla, and in between there is a cupula. So if our head is like this big arrow anteriorly, this big arrow represents the movement of the head. And the small arrows inside the canal, they represent the movement of the endolymph. So like if we turn our head towards the right side, so the fluid in filled in the semicircular canal, the endolymph will have the inertia of this fluid will move towards the opposite direction as of the head movement. The head is moving towards right, but the fluid will move towards the left side on the opposite because of the inertia of this fluid. So this movement is called as the ampulopetal because the direction is towards the ampulla, and this ampulla will cause, we call it an ampulopetal flow of the flow in the lateral semicircular canals, but it is in the right side of the lateral semicircular canal. So if our head is moving towards right, there will be an ampulopetal flow in the right lateral semicircular canal. At the same time, and ampulopetal flow in the right semicircular lateral semicircular canal will cause the increase of the firing rates in this canal and will our brain reads it like an excitation. At the same time, the left side of the lateral semicircular canal, there is a flow, we call it as an ampulofugal, opposite direction. So ampulofugal flow will cause the inhibition of the firing rate of the lateral semicircular canal. So movement of the head toward the right side will excite the same side of the lateral semicircular canal but inhibit the opposite side of the semicircular canal. This is because the lateral canals of the right and left are coplanar. That is what we are discussing about this thing. If we are talking about the superior semicircular canal, so the excitation of the one side of the superior semicircular canal will cause inhibition of the posterior semicircular canal of the opposite side and the vice versa. So this is because of the inertia of the fluid in the canals. The pressure on the cupula puts the pressure on the hair cells of the crista ampullaris. This results in the movement of the stereocilia. This is like a flow chart for easy understanding. And the increase or the decrease in the firing rate depending on the direction of the movement of the stereocilia towards the canocilium or away from the canocilium. So this is already we have discussed. Like this is a thumb rule which we all need to know. We need to understand this thing by learn by heart. Ampulopetal flow in lateral semicircular canal increases the firing rate and it's read as the excitation, whereas the ampulopetal flow in anterior or the posterior canals, they decrease the firing rate because it is in the this ampulopetal flow will cause the movement of stereocilia away from the canocilium. So that will be the inhibition of the these canals. Okay, so lateral for ampulopetal is excitation for the lateral canal and inhibition for the rest two canals, and vice versa. And the same time, opposite action is it happens in the coplanar canal of the opposite side. So this is all the same thing we have discussed in detail. The excitation of the ipsilateral canal due to the high velocity of impulsive head movement results in the inhibition of the contralateral coplanar semicircular. This is another rule. Always you need to remember by heart. The excitation of any canal is always stronger than the inhibition of the opposite canal. So stronger, the increase in the firing rate is much more stronger signal than the inhibition. So this causes the basic of the vertigo. Like if there is a difference between the firing rate of any because of the injury to the semicircular canals or the this thing, then like if there's a one side of the lateral semicircular canal is on excitation because of the injury, if there's a viral labyrinthitis or there's an acute inflammation, it will increase the firing rate. So excitation is always a stronger sensory input and inhibition will not be that much. So the difference will be too much. At the same time, if there is a dead labyrinth, there will be this excitation will be very much less and there will be a much more difference of the signals going from either side of the semicircular canal. So this difference is the signal. The brain judges that difference of the signals which are coming from semicircular canals of both sides, and it judges that the movement of the head is resulting uh, too much uh, as compared to the movement of the endolymph and the endolymph. So our brain judges as a vertigo. Now we'll discuss about the otolith organs. They are utricle and saccule. They are present in the vestibule. They judge the linear acceleration, the vertical acceleration for the gravity. It is judged by the utricle, and the horizontal acceleration is judged by the saccule. Again, the main sensory input is taken by the hair cells. The structure is same as of the semicircular canals. Just that these hair cells are embedded into the macula, and that macula is covered by a otolithic membrane. Here in this picture, we can see these are the hair cells, and these are the otolithic membrane, which is they are the calcium carbonate particles, and which are embedded. The movement of this otolithic membrane will result in the movement of the cilia, whether towards the kinocilium or away from the kinocilium, and that will result in the excitation or the inhibition. The basic fundamental is same, just that the structure in this utricle and saccule is slightly different as compared to the semicircular canals. Okay, so little bit in details. This is the linear acceleration or the change in the position with respect to gravity. They are judged by utricle and saccules. There will be a movement of the otolithic membrane, a deflection of the stereocilia results in the depolarization of the hair cells, and uh, depending on the direction of the movement of this stereocilia, this the brain judges whether it is an excitation stimuli or it is an inhibition of the this thing. With respect to the semicircular canal, this is again another third uh, basic fundamental rule which we need to learn is with respect to semicircular canals, otolithic organs like utricle and saccule, they have got a neural activity which persists during static head tilt also, which is because of the gravity, because they are always at work. because our gravity is present always, and that is what these autolytic organs are always at work. So now the central vestibular system, this will be discussing in the next class. Thank you so much.